Good afternoon, or good morning, or good day to wherever you are listening in today. It's a pleasure to welcome Dr. Karyn Esser for our MHRC seminar this afternoon. Karyn is now at the University of Florida in Gainesville, Florida, and at the Institute of Myology there. Just take a moment to give you a little bit of background on Karyn before she shares her slides with us. Karyn got her master's degree at the University of Nevada in Las Vegas. She then got her PhD at the University of Michigan in Ann Arbor, and then did a postdoc at Children's Medical Research Foundation in muscle genetics. She started her career at the University of Illinois in Chicago. I think that's where we first met. I believe that's where we first met. And then went to the University of Kentucky, where it's part of the muscle center there at the University of Kentucky. Stayed for 10 or 11 years, I think. And then has been recruited as a preeminent scholar to the University of Florida, where she is, as I said, at the Institute of Myology, doing great work and really setting her work originally in transcription, translation of muscle hypertrophy. The control of muscle hypertrophy was really wonderful work, and now she's really developed a fine niche for herself, studying circadian clock and circadian rhythms in muscle, and really the world expert in the field. And so we're just delighted that Karyn is with us today, and I'd like to welcome you, Karyn, and invite you to share your slides with us now and tell us about muscle clocks in muscle and what it all means, really.
Well, thank you, David. It's a real pleasure to be here. Let me just make sure I get this correctly done. And oh, I forgot to go into, hold on one second. All right, so now we go through this. Do you have the slides mode or do you have the other side? Do you need me to switch screens?
I think we're good. I can see your slide and I can see you, and that's perfect.
We're good to go. All right. Well, thanks again. It's a real pleasure to talk to the group. You have such a lot of strength up there, and I mean, no pun intended with muscle talk, but that said, in the area up there at York University. So what I want to do today is I've tried to break the talk down into 3 sections. The first is very brief and just sort of an update with where we are and our understanding of the circadian clock, and in particular the muscle clock. The sort of main body of the talk is really going to be the story that we've been working on for several years now, through which we're now getting a handle on the links between muscle clock disruption, the transcriptional output from that, and how that can impact muscle strength, or in this case, if you disrupt your clocks, does that lead to weakness? And then I want to finish with, it's quite a brief story, and it's really a little bit of a, it's just some fun data that we've got from some of our models, and I just want to throw it out there because we're really quite excited. We think we've, sort of not by plan, but have identified some interesting things about, how muscle might matter for systemic health. And for some, I don't know, someone like me that's been in the muscle field for so long, and especially working now in the last, 15 plus years in medical schools, skeletal muscle usually doesn't get much attention. It's not the heart, it's not the brain, it's not cancer. And so I've always been on a soapbox that muscle really matters more than our medical colleagues really recognize. And so I sort of feel pretty excited about the possibility of having a model system to ask some of these questions. So with no further ado, so the brief review of circadian rhythms, because I think first off, I didn't start in this field. As you noted, I started in transcriptional regulation of slow isoform genes, looking at signaling pathways and hypertrophy. So for those of you that don't know me, my venture into circadian rhythms was really one of the wonderful sides of science. It was an accident. It was serendipity. It was first generation gene array studies in the early 2000s. And it was a topic or this concept that there are these clocks in muscle just drew my interest and attention. But the field's changed a lot. So what we know about circadian rhythms is that we still do. I mean, we've known this for about, I don't know, hundreds of years, that they're approximate 24-hour patterns, and we can see it in physiology, we can see it in behavior, so things like sleep-wake cycle. We can see it in our biochemistry and metabolism. We can even see it in acetylation of the genome. But the feature that defines something, so it's not just that it happens over 24 hours, but this pattern has to happen in the absence or independent of any information from the environment. So that is the feature that makes circadian rhythms sort of unique, and that's the sort of intriguing aspect of circadian rhythms to me. So how do you get this intrinsic rhythm? Well, so we all have clocks, and it's not just in your brain. We have clocks everywhere in our body, in every cell. And sort of uncovering some of the initial mechanistics work was done in Drosophila, with obviously the power of the genetics there. But the reality is clocks are conserved across living organisms. So virtually everything, going from plants to bacteria to animals, everything has a circadian clock. And then this was the focus of the Nobel Prize in 2017, which always, let me tell you, it helps the field a lot. The point, I think, when I first got in the field that was really important is that this molecular clock, this mechanism exists in every single cell. So, and it works. So the clock just runs. It doesn't rely on the sun. It doesn't need a cue from anywhere. The clock is just this mechanism that I'm going to talk briefly about that just runs. And so again, I came into this field as a physiologist, and classically we think about stimulus and response. This is different, and it took me a little while to wrap my head around it. The clock is really an anticipatory mechanism. So it's not waiting for a stimulus, it's actually anticipating what it, what it thinks is going to be a stimulus that's going to come with, let's say the active part of the day. But we do have these clocks, but they are adaptable, or they are entrainable. And what that means is they get information from environmental time cues. Now, the cue we know the most about is light. That's been the most well studied. There's a lot of really interesting data about how that information is processed and how it manages shifting the clock and phase. But over the last 10 years, it's become very apparent that things like time of feeding, so when you eat, and again, an area that my lab is working in is sort of exercise or muscle contractions, and thinking about these as cues that the muscle clocks are listening to and adjusting the phase of their clocks depending on when those contractions come. So the clocks are adaptable. We're not going to talk a whole lot about that in the talk today, but it's important to know. And lastly, and so I'm having, so one of the things that we know that, well, we are learning is that there are a lot of conditions. So aging is one, but a lot of chronic diseases now have been looked at, and one of the correlates of these conditions are that the clocks in a variety of different tissues, ranging from your brain to your muscle, to your heart, to your skin, to your immune cells, these clocks are disrupted. And so it has raised real interest in whether we can target the clocks to actually improve things like quality of life with aging or other disease features in type 2 diabetes, for example. So that really gives you a sort of a brief, hopefully, overview of kind of where the state of the clock field is. Now, the timing mechanism that's involved in keeping in this 24-hour is in this cartoon that I've got on the slide. And so this is, I think of this as the primary timekeeper. And so it's really, it's quite simple. It's based on, there is a transcriptional phase, a translational phase, so you need the proteins, and then there's a feedback, and it's a negative feedback loop. So we start with the transcription that's regulated by these two transcription factors. One of them is called Bmal, and one of them is called clock. These are BHLH pos domain transcription factors. They heterodimerize. And for those of you that have thought at all about muscle transcription, they bind a very familiar site. That's an Ebox. And so they bind to these Eboxes upstream of another set of genes called the periods or the cryptochrome genes. They transcribe these. These are translated out in the cytoplasm to protein form, where they form multimers. They can be post-translationally modified. They will then translocate back into the nucleus. And when they get back in sufficient quantities, they will serve to inhibit the transcriptional activity of Bmal clock. This process takes approximately 24 hours. And sort of simplistically, we think about Bmal clock being the positive or turning on things, and then period cryptochrome being the negative limb to inhibit that. And that fundamentally is the clock, and how this works was actually the focus of the Nobel Prize. Now, biology is never that simple, and I'm not trying to put this on here to sort of, but because there's a lot of fundamental stuff that's the same. And in fact, this slide is now a good 8 years old. But if you can focus within the red circle here, you'll see the blue ball and the green ball here. These are Bmal and Clock. And as I had in the previous slide, they bind Eboxes to drive period transcription cryptochrome protein. And here you go. Here's your timekeeper. That is the same. We now know there's an accessory loop that's made up of the reverbs and the roars, and these will serve to interact with Bmal particularly, and also bring some external information into the clock. But and we're not going to talk much about this. This is sort of thought of as a stabilizer or a modifier loop. But I think the most important part for my talk today, and just generally, is this last part, which is the clock output. So it turns out that the Bmal clock transcription factors don't just bind to, periods and cryptochromes. They actually can be found all kinds of places across the chromatin. And so generally speaking, when people have gone in and characterized circadian transcriptome across tissues, they've identified, and it varies depending on the statistics you apply and your criteria, but there are a number of tissues, liver and kidney come to mind, that have 3,000 or 4,000 genes that are oscillating over a 24-hour period. Some tissues are a little bit less, but needless to say, this is not just a 1, 2, 5, 10 gene kind of program. We're talking thousands of genes are regulated in a daily manner. And so before I get into the sort of nuts and bolts of some of our, well, this is our work as well, but one of the earlier issues that I came upon when I started in this field was I had a number of well-meaning colleagues, but they didn't believe that this actually worked in muscle. And so they just thought it was just some kind of noise. These things were going up and down, and Karyn, what are you doing? And so one of the most important experiments we had to do was to try to do some experiments to demonstrate sort of the autonomous clock in muscle. And the way we did this is we started with what is considered the very gold standard circadian reporter mouse. So this was made in Joe Takahashi's lab in the early 2000s. And what they did is it's a knock-in mouse. So this is not a transgenic. So they knocked in the luciferase cDNA into the period 2 locus. So you have actually a chimeric protein. So it will make period 2, which functions as period 2, but it also has the enzyme activity of luciferase. So now what you can do is you can take the tissues out of these animals, you can incubate them with luciferin, and so they're going to make light. But that light and the expression of the light is going to reflect period 2 protein expression. And so what you can see over here on the right with these graphs are examples of different tissue explants, 3 different examples that were taken from this mouse, incubated with luciferin over about 10 days. And if you just focus on this one up top, what you can see is a really nice 24-hour rhythm, and that reflects period 2 oscillations. So now we have a system that provides a reporter, a valid reporter, that we can actually watch with bioluminescence technology and just ask whether these things are oscillating in muscle. So to do this, what we did, one of the experiment, we've done a few different approaches here, but what I'm sharing with you right now is going to be a video of a single fiber from the period 2 luciferase mouse. So this is just a single fiber we took from the flexor digitorum brevis muscle, and we put it in a dish all by itself. So remember, no neurons, no circulating factors. It's a cell, it's a fiber in a dish. And what you want to be looking for is changes in light across the fiber. And also remember, fibers are multinucleated. Each nucleus should be a clock. And so if you just watch the fiber over a 24-hour period, hopefully right now it's fairly dark, and now you start to see the light start coming. And each of those should be, I mean, we've not validated it, but each of those should be a nucleus, okay? And so it was data like this that helped convince my colleagues that, you know what, you can take a muscle, you can stick it in a dish, it doesn't need a motor neuron, it doesn't need anything, and these clocks will continue to run for days and days in this cell in a dish. All right, so that's the background. So now I want to get into, you know, what's going to happen. So you know, the other question, again, coming from a physiology background, is like, okay, well, we have a clock. Maybe it doesn't, you know, who cares? Maybe it doesn't matter. Maybe it's this sort of interesting timing mechanism that over the course of evolution really sort of lost its function in muscle, and it just sort of works, but it's not functionally relevant. So the next, I mean, you know, what I'm going to share with you now is really a summary of data that my lab has been fortunate enough to collect over several years. And some of this is unpublished, some of it's published. But the first way that we started this was to use a loss of function. So there were circadian mutant animals, and we had published some work with this back in 2010, both Bmal knockout mouse and a clock mutant mouse. But again, those were global mutants, and so you really couldn't ask very muscle specific questions. So this was while I was at Kentucky, we generated this human skeletal actin driving the Merck-Cremer transgene. So our Cree driver mouse that's very skeletal muscle specific. And then a group at Harvard made a floxed Bmal mouse. And then really all the rest of this, so I've got some PCR gels here in B, and then some protein gels in C. The PCR gels are just really there to say, okay, our recombination shows up across all kinds of muscles. So again, being a muscle lab, we don't just take one, we take a variety, and we got recombination in them all. And what I was really pleased with is that we did not see any recombination in the heart. And then we also did not see recombination if we did not treat the animal with tamoxifen. So one of the things I forgot to mention is this is a tamoxifen inducible system. So these animals get to be about 12 to 14 weeks of age. We let them get to adulthood, and then we treat them with tamoxifen for 5 days and then wash the tamoxifen out for a few weeks. And so we don't have any developmental issues with these animals, and we can control when we knock out Bmal. And then the protein gels just illustrate, so GTN is my shorthand for gastrocnemius. I've tried to go back and write it in, but if you see GTN, that's just gastroc. So we've knocked Bmal protein out in the gastroc, but obviously not in the liver. So that's all good. All right. So then you know, so then we wanted to say, okay, well, what are the genes that the muscle clock is regulating? And so this is just sort of really more like a brief method. I mean, it's not really methodology, but just I use it as a placeholder to talk about how we do some of the studies that we have to do in circadian biology. And so again, you know, we're talking about something that has a pattern. We now have this other axis, which is our time axis. And so we can't just take a couple of time points. We actually have to look at gene expression over an extended period of time. And I've put some data up that I've pulled from this really wonderful website. So if you ever are curious about your own gene, CircaDB, freely available, easy, as far as I'm concerned, really easy to use. Has muscle, has a whole bunch of other, has about 16 other tissues on there. You can always query the genes that you've got. But anyways, what are the things you need to do? Well, if you notice here on the x-axis, we've got, in this case, it goes from 18 to 64. So what this group did is they collected tissues from mice every 2 hours for 48 hours. So you went 2 full days. And so basically you have 2 full circadian cycles. And then they collected the tissues, isolated RNA, and this was done long ago enough. These are all gene arrays that were done. And this just gives you an example of the pattern of some of these circadian genes. So for Bmal, for example, part of the positive limb, it peaks at the end of the active phase of the mouse, or the end of the dark phase, early rest phase. And it's lowest at the end of the rest phase and the start of the active phase. And so you can see this repeated. And then illustrated down here is period 2. And what you might expect for a gene that works on the negative arm is that it is antiphase. So when Bmal is peaking, period 2 is low, and vice versa. So this really just gives you an idea of what it looks like to look at some genes that are circadianly expressed. The other point I will make is there's, you know, sort of there can be a 2, 3, up to 4-fold difference between the peak levels here and the trough levels here. So if, depending on how the people in your lab work, if you have some people that like are early morning people and some people that are late night people, you can be looking at the same tissue, but you can get quite different results on your gels. So just always something to keep in mind. So when we did this with skeletal muscle, I'm just going to cut to the chase. We detected, again, we didn't do the whole 48 hours because that can get exceptionally expensive, and especially in the early days of the microarrays. So we did a series of experiments where we collected tissue every 4 hours for, I think it was either 28 or 32 hours, and then ran the circadian statistics. And with a fairly conservative approach, we got over 1,200 messenger RNAs that are expressed in a circadian manner. These are just illustrated in the heat map over on the left, right here. And I mean, one of the things you can see is that the genes don't all peak at the same time. They peak at very different times over the course of the day. And then in the middle here, I've just pulled out some of the functional clusters that represented these genes. And in particular, what I'm going to be talking about today are the genes that are related with transcriptional regulation. So the idea here is the clock is actually targeting a number of transcription factors. One of those transcription factors is MyoD. And then the other thing the clock does target that I'm going to talk about today are some of the structural proteins in muscle. So I mean, there are a number of other people that have started to look at the links between the clock and metabolism. And there are a lot of links there. And that's also quite important, but that has not been really the focus of my lab. So I'm going to focus on the MyoD and the structural proteins here. But basically, you can find that almost all aspects of cell function are under clock control, which brings me back to this concept that the clock regulates what I would consider a very much a homeostatic transcriptional program. So what happens when we knocked out Bmal in the clock? And so this is just putting these heat maps side by side. So all the genes are here are represented in our muscle-specific Bmal knockout. And so, I mean, hopefully what is visually apparent is that we've basically disrupted the whole pattern of gene expression in the muscles of these animals. And it's important, and that's why I've got it in the title of this slide. So we monitored feeding activity, we monitored cage activity. Those things did not change. We're not changing the brain clock. We're just changing the muscle clocks. So this animal is feeding like normal. It's moving around like normal, but and we don't just turn genes off or turn on. So it's not like sort of we've really just muddied up the transcriptomic program. So this data is now 10 years old, or actually older really when you think about when it was done. But as I alluded to you, we had seen that MyoD came out in this first analysis of the circadian transcriptome. And I'm making an assumption, since this is a muscle group, that you're all familiar with the transcription factor MyoD. but it is a very important one, as it was the very first transcription factor that was ever identified that was shown to confer a lineage specificity when transfected into a fibroblast. So you could, the very first experiments were this, where they transfected MyoD into a fibroblast and they turned it into a muscle cell. And so that was really pretty, the very first transcription factor that had that capacity. We've gone on and we know a lot more about what MyoD does, its role in regulating a number of different muscle specific genes. So we were really quite intrigued. So that hopefully introduces you as to why did we pick MyoD out of a variety of different genes in there, because of its role in muscle. What I've got in these figures down below, so here's MyoD in a wild type in the solid line. And then in the open circles, this is taken from a mouse in which the clock gene is mutated. And basically the point again is we don't turn MyoD off by any stretch, but what we do see is a loss in the circadian oscillation. And then we've got some MyoD protein gels, and hopefully what you'll see there is that MyoD protein does oscillate. So this is just wild type tissue. This is not the clock mutant. But so not only does the message oscillate in wild type muscle, but the protein does as well. So again, conveying the concept that there is some functional effect of the MyoD oscillating in muscle. So again, going back to the history of MyoD, it's a basic helix, loop helix transcription factor. It binds Eboxes. And so I'm sitting in these circadian lectures and everybody's talking about Eboxes, and I'm like, but wait a minute, MyoD binds to Eboxes. So very simple question. Could these things actually be cooperating? So again, let's see, my transition slide I seem to have left that out here. So when we went to ask this question, we thought, okay, let's dig through our array database and find a gene that we know is, so A is muscle specific, B is oscillating in normal muscle, and C, we lose oscillation in the muscle specific Bmal knockout mouse. And for the purposes and then we're going to use this gene as our model to study. So the gene we identified, or one of the genes we identified, is the gene Titin-Cap, Tcap. It's also known as telethonin. And we'll talk a little bit about its function later, but as you can see, it's got a very robust circadian expression with the peak levels at the end of the rest phase, start of the active phase, and then it oscillates down. And when we knock out Bmal in these mice, what we see is we lose, not only do we lose the circadian expression of Tcap, but the expression of Tcap is significantly lower. So, and again, so where we started is just some very classic molecular biology. And we said, okay, first let's look at the Tcap-reporter and see if we can identify some sites that might be used by Bmal clock to regulate expression. So in the gray, a bunch of letters, but not too surprising, we found 3 Eboxes in there, or these two in red and this one right down here. And there had been work on Tcap-reporter before, so we had a hint that there were going to be some here. So we made our generic reporter vectors. So we took this promoter region, put it upstream of luciferase, and now we're going to start some transfection studies. First thing we had to do is figure out if it actually, so not just whether it's expressed in muscle, but now we've got, again, I go back to that, we've got this other domain, that's the time domain. So we had, I mean, so this is the wonders and beauty and challenges of starting a new field is we had to make the assay for this. No one else had done this, so we had to see if this was going to work in C2, C12 cells. So what you're looking at now, we'll just focus on these right now. So we transfected Tcap into C2, C12 cells. We differentiated them into myotubes. And then again, this is luciferase based. So then we gave them some luciferin, and then we've got a system that does real-time bioluminescence recording. So, and so what you're looking at are the circadian oscillations of the luciferase expression from the C2, C12 myotubes, and this is over 4 days. Okay, so one is, you know, oh, yay, it works, the C2C12 system. So C2C12s do have a clock. I'm not going to get into all that, but they do. They can be synchronized and they can be used for circadian studies. What we found is if we actually boosted MyoD by overexpressing MyoD, we actually could increase Tcap expression very significantly. You get a much bigger amplitude now. So anyways, what this established was that we had a model system to study both transactivation of Tcap expression, but also circadian behavior of the promoter, and does it drive circadian expression. So the first very simple experiment was just a very classic transfection experiment. So in this case, we're going to take our Tcap-reporter, and we're either going to give it MyoD or we're going to give it all three. So MyoD plus Bmal1 and Clock. And what we got was about a twofold transactivation of Tcap. And I want to point this out because one of the things I realized putting the slides together today, Bmal1 and Clock can upregulate Tcap some, but it wouldn't be, this is not, you know, this is more than the additive function. So this was our first clue that there might be some synergy going on between MyoD and Bmal1 Clock. So the next set of experiments that Brian did is he actually said, okay, what if so let's look at this. We've got a, you know, so we're looking at a 600-fold increase in luciferase activity when I put all three of them together. What happens if I put in, you know, MyoD, Clock, but I put a Bmal mutant in? Or I put in MyoD Bmal and I put a Clock mutant in, or I put Bmal Clock and put a MyoD mutant in, or or the combination of the mutations. And it was really quite profound. And this we actually weren't really expecting, but no matter which set of transfections, you virtually knock down the expression of Tcap. So I mean, we could, and I'm not going to get into the difference between this or this or this. The main point is these are all way below this and even lower than this. So what this was telling us is these things are not working in isolation. These things are actually likely working as a complex. So then after doing, you know, so we played with mutant forms of the transcription factor. So then we went back to the promoter and said, well, let's start mutating some of these putative E-boxes that we see in the promoter and see which one or ones are important for expression. So again, here's our, this is just MyoD alone. This is plus Bmal Clock and MyoD. And whether you mutate this proximal E-box or the one that's a bit more distal or the most distal E-box, any of these E-boxes, we don't lose the MyoD ability to activate, but we do lose the ability to get the synergistic expression from all three. So, so the slide before was telling us that all the proteins were needed. What this data is suggesting is actually that more than one E-box is needed, that these things are and they may be very much using multiple E-boxes on this site to function as a transcriptional activator. So now what we're going to do is we're going to go in and we're going to do mutation of the E-boxes is what I've got. Is that right? Yes. So so in this case right here, so this looks basically like wild type, and that's pretty much what it is. So this is a mutation. We've put a point mutation in E1. And then, so we still have circadian expression. But if we put a mutation in either this E-box or this E-box, so either of the two E-boxes under that heading of tandem E-box, as you can see, we lose basically a combination of things. We lose expression, so the expression is generally lower, but we really lose circadian expression. So the pattern is completely lost with either one of those. So again, suggesting that there is some kind of complementary requirement for these two E-box sites that sit very close to each other. So they're only something like 7 or 8 nucleotides apart. But we think this is going to be an important sort of home for MyoD, Bmal1 and Clock. So this is all sort of molecular biochemical assays. So then I was fortunate enough to have a new postdoc come to the lab and say, oh, wow, well, I've used sort of BiFC techniques. Well, BiFC, I'm like, well, what's BiFC? So as someone that hasn't used a microscope a whole heck of a lot. So these are bimolecular fluorescence complementation assays. So basically what you're doing with the BiFC is we're going to take either clock, so it's sort of pointed out here in the title of each of the figures. So in this case, we've got part of the fluorescence on the clock and the other part on Bmal. And the only way you're going to get fluorescence from these cells is if the Bmal clock are close enough to each other that these two pieces can interact. So what the interaction tells you is that these two proteins are in close approximation. And so when we use just, so a clock and a Bmal in this assay, hopefully what you can see, this is in a C2, C12 cell, is that the whole nucleus is lit up. And it's not just one site, it's across the whole nucleus, which is, again, I mean, we're not going to go into it, but too much, but basically Bmal and Clock bind to 4, 5, 6,000 sites based on ChIP-seq data sets. So this is very consistent with what we've known with Bmal Clock in across the nucleus. This is just our negative control between Bmal1 and 2 that we know don't work together. Now, if you go over to C, what you'll see is Clock and MyoD. And again, what you see are some beautiful, fairly large, but also several puncta within the nucleus. So again, it looks like there's some very big organizing centers across the nucleus where MyoD and Clock are working together. And then over in D, this is just the same thing, only with Bmal and MyoD. And so hopefully what you can visualize now is very consistent with all of our biochemical data, that Bmal and Clock and MyoD really are neighbors. They're collaborators, they work together, they can be found throughout the nucleus and muscle cells, and they can work together to regulate transcription. So as I alluded to, another set of experiments that my lab has done is to perform some chip sequencing studies. So in this case, we had done a series of MyoD chip-seq experiments that was published in the eLife paper in 2019. The Bmal Clock chip-seq data is unpublished to date, but I want to share a little bit of this. So what I've talked to you about, we use Tcap, so we have clear evidence that MyoD, Bmal, and Clock regulate Tcap. Well, Tcap is this protein, it sits on the Z-line, it plays a role in gluing Titin down into the Z-line structure. So it has a very important structural role in the sarcomere. So when we were also looking over the data set, what we noticed, there was a gene that was bound by MyoD, Clock, and Bmal. And this is the visualization of the ChIP-seq data. And this gene is called RBM20, or RNA binding protein 20. And here you can see the shared binding sites. And I've provided a little summary of our ChIP-seq data. So we get Bmal Clock sharing, and I mean really sharing peak binding at about 6,000 genes in adult mouse skeletal muscle. And then when you overlap that with the MyoD data set, we've got about 3,000 genes that are in common. And our criteria, just for anybody interested, is fairly stringent with a peak call of about 14 and a false discovery rate of 0.001. But anyways, as I said, another of the genes that was intriguing to us is RBM20 was the target of the clock binding. Because this has, actually it's been studied more in the heart, but it has, mutations in RBM20 are linked to a variety of different cardiomyopathies because you get Titin missplicing and structural problems. So simple question is, do we see changes in RBM20 expression in our muscle-specific Bmal knockouts? And the simple answer is yes, we do. So this is just some messenger RNA data. We have significant reduction in RBM20 message And I didn't have time to pull up the protein data, but in fact, the protein data is even more pronounced. So this difference is somewhere around 25 to 30%. The protein data would say it's more like 50% down in the muscle. So then, okay, well, if RBM20 is down, then one would predict we're going to have Titin splicing issues. So back to sequencing. So now, of course, it's a lot easier and cheaper to do RNA sequencing. And I have some wonderful colleagues here at the University of Florida that can also were really helpful with helping my graduate students. So this was done by Lance Riley, learn the pipeline for doing splice analysis. But the cartoon, and again, Titin is huge. So so one of the things I was thrilled with is, one, there was a difference, and that's highlighted in this red box. So if you just look at, so this PSI stands for percent spliced in. So what we would expect with RBM20 levels going down is we would actually have inclusion of more exons, and in particular in this region. So everything, it worked, I mean, it came out. So the fact that the red line here is above is indicative of the inclusion of additional exons in the Titin message. And fortunately, for me anyways, was that we really saw no other effect of splicing across the whole length of the Titin molecule. So that was a relief. So it's consistent with RBM20. We know the region it's in, sort of the I bands close to the PEBK region. And it's an area that is commonly spliced, and in particular, it is a splice target of RBM20. So then, okay, we've got changes in the splicing of the message. Do we see it? Can we see the effect in the protein? Now, Titin's huge, so you don't just run polyacrylamide gels. So to look at Titin protein forms, you have to use a vertical agarose system, because Titin's the stuff that gets stuck in your wells at the top from muscle. Anyway, what you have is it is a cropped image, so this is the wild type. You have mostly the bottom band with a little bit of the top band. And what we see consistently in our muscle-specific Bmal knockout mice is we have a higher contribution of what I'm going to call is the long form. And that's illustrated in the histogram here. So the point with this is that, so A, the changes in splicing are being followed by changes in the length, the protein form of Titin, so that's all consistent. But the other part of this is that it's not like, so we're actually creating a greater mixture, right? So in wild type muscle, we're maybe only 20% of the long form, but now when we knock out Bmal and we reduce RBM20 by about 50%, we're now up to about 40% of the long form in the muscle. So, okay, who cares? What does that do? Well, a lot of work from a lot of really good labs are now teaching us that Titin length can impact sarcomere length. So one of the things that Lance did is he did some longitudinal sections of the muscles and stained them with alpha-actinin. And so that's, you know, this image you can find in lots of books. So you've got alpha-actinin, which marks the Z lines. And then in this case, he's got DAPI staining. And so he went in and measured average sarcomere length. And then he stained the Bmal knockout muscle, did the same thing. And lo and behold, there's no change in sarcomere length. But what he did notice is that the variability was much, much greater. And in particular, there was a bias to longer sarcomeres in the muscle-specific Bmal knockout, okay? So again, we've got a mixture now of long and short. I mean, this would suggest that you're getting inclusion of more inclusion of these long forms into the myofibrils or the myofilament system, and you're resulting in sort of this mixture of sarcomere lengths. The other thing you may notice is the staining of the Bmal knockout is not as robust, or it's not as vibrant. This is something I'm really quite interested in, and it's not because the alpha-actinin's not there. They have plenty of, they have normal levels of alpha-actinin. This is actually an issue of being in plane. And so we are also, there's some other things going on that we, right now, it's one of the models we're testing, is that there is actually some loss in orientation or some modification in orientation of the myofibrils. So now when we go through with confocal, we can never get that sort of vibrant, all in one plane staining of alpha-actinin. Anyways, that's sort of a nuance that I'm really excited about. At the end of the day, I'm a physiologist, so who cares whether your structure is off? Well, muscles care. Muscles really require, you know, the structure to be, how are we doing here? The structure to be in place. And so one of the things we found is that when we knock out muscle-specific Bmal, grip strength goes down, and then really the gold standard is in B. And so this is an ex vivo force frequency test. And so you're looking at frequencies on the x-axis and force normalized for cross-sectional area on the y-axis. And hopefully, I mean, really the initial part looks quite similar, and that would suggest the calcium handling is probably okay. But where we see the loss is out here at the tetanic force, maximum isometric tetanic force. We've got about a 20% reduction there. And again, remember, these things are normalized for size. So this is not because they're smaller, it's just they don't work as well. And so I'm not going to go in. This is just another feature of the assays we've done. And so there are a variety of mechanical properties that are changing in the muscle, and there's still a lot to be learned here. So to summarize this part of the talk, what we've done so far is really the studies where we've done loss of function of Bmal only in skeletal muscle. What we've used these kinds of experiments to do is to help us understand what that daily transcriptional program is from Bmal clock, and in particular, this concept that Bmal and clock are actually working with MyoD to contribute to the regulation of a very muscle-specific circadian expression program. And one thing I didn't mention early on is that, as I said, every tissue has a circadian transcriptome. So liver, heart, you know, fat, any, you know, you name it, they all have circadian transcriptomes. But if you compare them, there's very, very little overlap. So there's, you know, the only thing that is in common between the circadian transcriptome of skeletal muscle and heart, for example, is primarily the clock genes. Very, very little else is in common. So one of the mysteries of the field is, how do you start with a ubiquitous program, Bmal clock, period, cryptochrome, Bmal clock bind genes, but yet get very different tissue-specific output? And so what we think we're learning in skeletal muscle is that we would propose that this is done through tissue-specific transcription factors. So in our case, it's the clock factors. They're going to work with muscle transcription factors, in this case, MyoD. And that way, what the clock's going to be doing is reinforcing the genes that the muscle needs to maintain health. So then, obviously, you lose Bmal or you do something to disrupt the clock. You're not going to just change one gene. You're not going to just change timing. You create a whole cascade of effects that will have implications across both structure, function, and, as I said before, metabolism for sure. We've identified a set of downregulated genes that include Tcap and RBM20. These things I think are very, well, we know they're very important for muscle structure. And then along with that, we have structural changes, we have functional changes. So then if you start thinking about, well, why does the muscle clock matter? Well, we want our clock to work well. And you know, maybe that weakness you have when you have jet lag, maybe part of that is, you know, the clock's out of whack and some of the things just aren't in the right place at the right time. And so, so what we would, any type of chronic disruption of the muscle clock, we would predict would lead to some kind of muscle weakness. All right, in the last few minutes, oh, I gotta do this real fast. So it is a short story, and there's no answer, so I'm gonna leave you hanging. I'll just tell you right now. What about the system? So one of the things that always happens in a lab, and I'm sure David has his share of stories about this, is someone does an unplanned experiment. Okay, so here's one of the ones from my lab. So one of the people in the lab took a cohort of the muscle-specific Bmal knockout mice. They were they were in the animal facility. Somebody either forgot them or whatever, but they got to be 8 months. And then there was an associated so there were 7 muscle-specific Bmal knockouts. There were 5 of the age-matched vehicle controls or wild types. And so then they threw them into the light boxes and did what we call a circadian stress test. What this is we will shift the clock 6 hours every 4 days. So it's like someone coming in, let's see, so I'm not really good at thinking on the fly here. So if, let's say you go to bed at 11 o'clock normally and you get up at 6 or 7. Okay, so now what we're gonna do, you go to bed 11, I'm gonna turn the lights on at 2 in the morning and you gotta start your day, okay? So we're gonna do that to you. And then every 4 days, we're gonna repeat it. So it's pretty stressful. But it was more stressful than I thought, and particularly with the muscle-specific Bmal knockouts. They started dying. And this was like, okay, I mean, what did they die of? No clue. This was, someone walked down, oh, we got another one dead. So it's an easy assay. They're either alive or dead. But it was only in the muscle-specific Bmal knockouts. We did not see any mortality effects in the wild type animals. So that was sort of like it sat in my head. And then it's triggered thought. So I had known very early on, if you just look at, so the Bmal knockout mouse has been proposed to be a model of accelerated aging, in large part because it dies early. So it dies its average lifespan is 37 weeks. And this is a mouse that we had used many years ago and had done some muscle function on. And like our muscle-specific Bmal knockout, the muscles, as not surprising, the muscles in the global Bmal knockout are also weaker, or the specific force is reduced. And so this sort of led to why not experiment, which was, okay, if we have our muscle-specific Bmal knockout, it's dying early if we put it in a circadian stress test. The Bmal knockout dies early. What would happen if we actually rescued Bmal in the Bmal knockout? Would we improve the lifespan of those mice? And again, part of this too is being at the University of Florida, where the use of adeno-associated viruses is, it's sort of like going to a vending machine. They're everywhere. So it's been a very big tool in the research community down here for many, many years. So it's really easy to access. So what did we do? So we have the wild type, we have the knockout, and now what Miguel's gonna do is he's gonna inject the knockouts with an AAV that's going to be systemically delivered, but we've put the muscle-specific promoter in front of it. So the virus will go everywhere, and it's a serotype 9, so it's something that the muscle will take up as well. But the promoter should direct, and did direct, muscle-specific expression. And since I'm trying to keep this short, the punchline is, yay, guess what? The animals live longer. So this is just a survival plot here. And so the purple is the Bmal knockouts. And like all the publications that have been done with this mouse, our average lifespan, or median lifespan, was 38 weeks. So we're right on there. As you can see, I mean, we had a lot of mice, so we weren't gonna fool around with this. We were either gonna show it or not. And when we took the knockout and introduced the AAV, we did have a few deaths, but there was a significant improvement in lifespan. So again, your muscle matters to your system. Surprisingly, so one of the things with the Bmal knockouts is their body weight is fine early on, but then about 12, 14, 16 weeks of age, they stop growing. The other animals start getting a little bit bigger. We had, the AAV had no effect on body mass. We do see a change, and again, due to time here, we'll just look at the total activity. So these are just wheel cages, and so this is a voluntary wheel activity. So when we rescue Bmal in the muscle, we increase the amount of time and the activity they do on the wheels, but they're not circadian. So they're active, which is great, but they don't follow a day-night pattern. No change in fiber type, so I don't need to, that's that. I mean, the fiber type of the TA muscle in the knockout animal is slightly different. It's slightly faster than the wild type, but the AAV does not create any fiber type changes. And the big surprise actually was fiber area was smaller with the AAV. So this I absolutely did not expect. This one I'm still kind of like jaw dropped and going, wow, what the heck's going on? So we're increasing longevity, but we're making the muscle a bit smaller. But interestingly enough, when we sent the muscles over to Elizabeth Barton's functional assessment core, we have rescued specific force in these animals. So this is where I'm going to leave you and summarize it right now. So what these results, I'd like you to just, I don't know, I'm just excited about it. They provide, because now we've got a model. We've got a mouse model. We've got a mammalian model system where we can ask questions about and try to identify either the molecules and the pathways from which the healthy muscle is contributing to a more resilient system. And whether that's through metabolism, whether that's through the immune system function, some kind of combination thereof. I mean, honestly, I have no clue right now. This is something we're just jumping into. I'm just excited to have the model working. I was surprised. We were all surprised that the rescue of Bmal in the knockout didn't do anything to increase muscle mass. But again, this has been something, I mean, my lab used to study muscle size, and I have to say over the last few years, I'm not saying muscle size isn't important, but there's something that, you know, healthy muscle strength is telling us that we just don't know exactly what to interpret. It's good, right? So you really want to maintain muscle strength. Mass is important, but with aging, with cancer cachexia, with a variety of chronic diseases, strength usually changes earlier, and it usually is a much bigger magnitude than the muscle mass changes. And so there's something magical when strength is preserved. Anyway, the most important slide is my Florida folks. And so I've been really fortunate to have a good group of people. Some of these have actually moved on, like Lance now is up at Vanderbilt doing a postdoc, but it's been a real joy, and I have some wonderful colleagues and collaborators. So thank you very much for hanging out with me, and happy to take any questions.
Karyn, thank you. Thank you very much. That's a great talk and great background and great data on the clock system. I'll ask a couple of questions. I'll ask the participants to please use the Q&A forum to ask your questions. And I have a couple lined up here. I wanted to just ask to start. Well, first of all, I love that vending machine for AAVs. That's we could use a vending machine like that right here as well. So that's nice, nice attribute of being in Florida. You know, the Bmal phenotype seems to be stronger than the clock phenotype. Is that true, or have you looked at the clock mouse versus the Bmal mouse?
Yeah.
You know, we had the clock mouse a while back, and the phenotype wasn't that severe. So
yeah. I mean, because I remember you worked with the clock, there's a clock mutant mouse. So the clock story is a little complicated, and no, again, I mean, so the problem is with these circadian experiments, you have not just one, but you got to get all these darn time points, which increases, and then the cost of everything. So we have not gone into doing, let's say, a clock specific knockout. Now, the clock mutant has absolutely, it has weakness. I don't think the longevity is affected. It seems to exhibit more metabolic phenotypes. But again, that clock mutant mouse, and I think that's the one you used, is a hypomorph. So it's a mutation in the clock gene that if you leave those animals under constant conditions, their period length is actually, I think, 28 hours instead of 24.
Mm-hmm.
So they have a slower clock. But the other part with those mice is they do entrain. So they're kind of hard. They are hard to, they're a little more complicated to interpret. Let's just put it that way.
Yeah.
There are a lot of people. So people have done a clock knockout. There's another, there's another gene called NPAS2 that seems to, if clock's not there, it'll take over. For whatever reason, Bmal1 doesn't have something that will compensate for it. It's the only gene, so that is part of this timing mechanism that's vulnerable, and in that nothing else takes over if it's gone.
Okay. Yeah. Okay. Let me get to a couple of question and answers here. Chris Perry is asking, could the lower force in the Bmal knockout also be due to changes in factors other than the sarcomere, like fiber type shifts? Well, it looks like you had no fiber type shifts, but what about glycogen or something like that?
Well, I mean, so specific force is, I mean, one of the things about specific force is it unless you've got some kind of really wild or really sick metabolism, that should not be affected, right? So this is going to be, this is not fatigue. This is, a measure. So when we do the force frequency curve, they rest between each bout of generation. So it's very different from, let's say, a fatigue test or a fatigue run. And so generally speaking, when people think about specific force, metabolic factors are really not very causative for reductions. Now I'm not going to say, and again, so this was also done ex vivo, so this is not a neuromuscular junction issue. But I'm not going to say it's just the sarcomere. I don't, as much as I'd love it, because I love sarcomeres, I think that's bad biology, so I'm not going there.
Okay. Next question from Tammy Martino at Guelph.
Oh, hey, Tammy.
Yeah, great talk. She says, I'm wondering whether the AAV rescue effects on mortality would be different in males versus females.
Awesome. We don't know. Well, we do know a little bit. So I take that back. So this is all unpublished. So we did do females. It turns out the female Bmal knockouts have a slightly longer lifespan. So they live, so their average lifespan is closer to 40 weeks. So what that means is, I mean, we saw a trend for the AAV in the females to improve lifespan. But we had ended the experiment at 40 weeks because we were actually initially just trying to mimic the Kondratov paper, which had run to 40 weeks. We actually, fingers crossed, I think got some good news today, which means we actually are going to do these experiments properly. And so and one of the things, actually, I probably want to talk to you because I am really interested to know whether there's some cardiac specific effects in these animals.
Just on top of that, are there sex differences in the clock at all? I mean,
I don't know. I mean, Tammy's on. She may know more than me, but I mean, this was. I'm smiling because again, I got good news literally today. So this is like a happy day. I should have a beer right here. But one of the things I had in one of my aims was that there are very few. Almost all the circadian transcriptomes are male. There are some that are mixed male and female, but they're, some kind of combination. As far as I know, there's no comparison male to female with circadian transcriptomes. And this is something that we are also going to be doing because I think it's fundamentally important. We don't know, as far as I don't know, and it's not in skeletal muscle, whether there is something like that in another tissue. If Tammy knows, feel free to chime in.
Okay. John McDermott asking.
Hey, John. Got some.
Since you showed that MyoD seems to be a collaborator in the clock, I wondered if the muscle clock is modulated in MyoD knockout animals.
Yes. So well, I mean, so what we have done, actually, it's funny, having a flashback. John shared a plasmid with us back in our early Chicago days, a MEF2, I think. Anyway, so what I can tell you is that a couple of things. So when we had used David Goldhammer's mouse, where he knocked out the core enhancer of MyoD, the CE knockouts. So the core enhancer is about 23 kilobases upstream of the MyoD start site, and it had been shown to be important for proper MyoD expression during development. Turns out that's where clock and Bmal bind MyoD, is in that core enhancer. So when we look at MyoD expression in those animals, it is damped, so it does not oscillate anymore. Now, what we also learned when we looked at those animals is the expression of Bmal is also affected. And so that is yet another one of the arms of our studies is, is that there is a cooperative sort of, so the arrows go both ways. MyoD, or Bmal and Clock, regulate circadian expression of MyoD. MyoD seems to be an amplifier of the clock. It doesn't change phase of the clock. It doesn't, but it will act to either increase, if it's available, it acts to increase. And is that what did I answer the right question, or did I answer the wrong question? Is that what you asked?
I think we got a couple of questions on MyoD. So Mike Connor is also asking about MyoD, about its levels during development and proliferating and differentiating cells. My impression was MyoD goes down as the muscle matures. Is that correct?
Yeah, yeah. No, I mean, so this has been an interesting. So there's a lot to the MyoD story. Let me just start with the MyoD knockout has set our field. I mean, it was wonderful. Mike Rudnicki, generating this MyoD knockout. But I think everybody was really surprised when it was born and it was normal weight and the muscles looked normal. Now, the muscles in the MyoD knockout aren't normal. So the specific force in the MyoD knockout is reduced. So our lab has shown that, as has Scott Powers, as had, I think, a couple of other labs. So the muscles in that MyoD knockout are not normal. The other thing that is really important, but you'll never find it in a paper, is there are at least 3 labs so the MyoD knockout is on a BiFC background. There are a number, there are 3 labs that I know of that have tried to backcross the MyoD knockout onto the Black 6 background, and my lab's one of them. Rudnicki's lab is another one, and I think David Goldhammer's lab is the other that I know of. You never get them. I mean, if you get some pups, they die before you wean them. We didn't even get pups, so there is something, it's a lesson, I guess, for the trainees, and it was a lesson for me in terms of mouse background and how that might affect things. But we have just, we're getting, David Goldhammer made a Phlox MyoD. So we are actually going to do an adult MyoD specific knockout mouse to ask some of these questions. Now, MyoD does go down with development or maturation. But the other thing, if you actually go back to our original paper, the peak of MyoD occurs in the middle of the dark. So it occurs at like 2 or 3 in the morning, and it's lowest during the daytime. So for most of us, when we were looking for MyoD in muscles, we're looking for it at its lowest point during the day. If we happen to have our animals on reverse light cycle, we might have had a very different impression.
Yeah, I guess I was thinking that if MyoD pretty much disappears in adult muscle, how does it work in concert with Bmal and Clock in an adult mouse? But if its expression is high enough, then I guess it could.
Yeah, like I said, we've done ChIP-seq with MyoD, so we had access to Steve Tapscott's antibodies, because one of the things is getting good antibodies for ChIP-seq, and MyoD is not trivial. And I mean, it's a numbers argument. We didn't isolate fibers, but that said, the amount that we were able to get And the genes that they were targeting are not things you're going to find in a satellite cell or an endothelial cell or a fibroblast, right? You're not, you know, MyoD is not going to, and so there's clearly sufficient amounts there. And so I think, I do think the MyoD, the concept of that MyoD might be doing something in an adult muscle kind of got lost in some of the noise.
Mm-hmm. Okay. So Karyn, just a couple of questions from me on the Bmal knockout again. I was just shocked at that presentation of those gene products on that I don't know, heat map or whatever it was when you knock out the disruption that occurred in the Bmal knock was just remarkable, I thought. Is that true? Am I?
No. I mean, honestly, it is. I mean, and it's sort of, you know, on one hand, it was very exciting. On the other hand, it's a little overwhelming. And I think, you know, for someone like me that can be distracted a lot, it's sort of like, okay, where do I focus here? Mm-hmm. And like I said, most the people that have come into the muscle field, the muscle clock field in particular, are primarily focused on metabolism, and there's a lot there to study. But I you know, my metabolism training is not. I mean, yeah, I'm more of a physiology, structure, function kind of person in transcription. So we, you know, as I was looking at things and sort of seeing who else was coming into the field, I thought, you know, I'm going to stay away from things like, you know, regulation of mitochondrial function. So, you know, fission, fusion, all kinds of stuff. There are parts of those pathways. Yeah. The glucose handling pathway. So both my lab, Schiaffino's lab, and others now have shown in the Bmal knockout, glucose uptake is basal levels are fine, but either insulin or contraction-mediated glucose uptake is just nothing. And some of that is GLUT4 levels are down, but you also have other components of the insulin signaling pathway that are affected. There's also glycolysis. So you're really, at least based on the limited metabolomics we've done with these mice and these muscles, you're really pushing, the muscle is now forced to use primarily fat as a fuel. And so there are, and actually I always joke around because my lab can genotype by smell, right? So we can open the animal up and you can smell, and I assume that's a metabolic byproduct.
I see. That's a useful quick test, though, isn't it? You could do that.
Yes, it is.
My one last comment on this is that the Bmal knockout, when you put them on the running wheel, their activity levels was very, very low.
Yes.
Yeah.
Oh, yeah. So, no, and these are, yeah. So yeah.
So if you're going to look at metabolic things like mitochondria, you have to certainly take into account the fact that they hardly have any activity compared to the wild type. Yeah.
Absolutely. Absolutely.
Yeah. Okay, folks, any other questions for Karyn before we allow her to go for that beer that she was asking about just a second ago? Any other questions? Not seeing any. Karyn, please stay on the line with me. I want to thank you very much for coming to us and sharing that great information. Folks, the webinar seminar is over now, and appreciate you coming and listening to Karyn, and we'll see you again down the road when we have our next one. Thank you very much.
Thank you. I guess I could have done this.